Hello and greetings from Ireland. My name is Dr. Ciara O'Leary from Inland Fisheries Ireland. Myself and Dr. William Roach are the leads on Work Package 6, the title of which is the Biological and Ecosystem Services, Data Collection and Case Studies. To put WP6 into context, we are collecting new data and collating existing data for the case study areas for the DiadES project. We will feed into the ecosystem services valuations and the new species model, as well as the interactive atlas and policy guidelines. Our deliverables under this work package are 6.1, which is a manual on the methods used to collect data for WP6. 6.2 is a database containing all the information gathered under the DiadES project. And 6.3, which is our input from the case study locations into ecosystem services or work package 4. Apologies if you've seen this already today, but here are the locations of our case studies and our partners. This is to give a flavour of how widespread our data collection is and the importance of the DiadES project in gathering data at this international and spatial scale. The different partners from the field of ecology and economics. The target of our data collection efforts are diagamous species, with a focus on the lesser known or data deficient species, such as shad, the thin-lipped mullet, sea lamprey, smelt, and the European eel, along with the more famous salmon and sea trout. Our first deliverable was to compile a manual on data collection methods for all partners. This was to ensure a standardization across areas to allow a comparison of the results. The topics covered are sampling for environmental DNA, extracting otoliths from microchemistry, but also managing the data. We are sending samples from multiple institutes to different laboratories, so data management is very important, and we are using Collect Science for this purpose. This manual is available on the DiadES website. Under WP6, we are collecting new data. The first is an environmental DNA analysis, and this is being undertaken in 5 areas: Ireland, the UK, the Giron catchment, northern Spain, and the Ulla catchment in Portugal. The target species are sea lamprey, shad, sturgeon, thin-lipped mullet, and smelt. Secondly, we're looking at otolith microchemistry for shad and thin-lipped mullet. Samples from Ireland, the UK, 2 locations in France, northern Spain, and 3 locations in Portugal. Our third analysis is Shad Hybridisation, which is taking place in the same catchments: Ireland, the UK, the Giron, northern Spain, and the 3 Portuguese catchments. So what is environmental DNA? While fish are swimming around their water, they're leaving traces of their presence as DNA. This can be skin and scales, mucus, eggs, and feces, which we won't dwell on. By taking water samples and analysing for the presence of this DNA of our key species in DiadES, we can detect their presence in areas where it is very difficult to survey using other methods such as netting. This data will allow us to look at the ecology and distribution of diagamous species in our case study rivers, in our estuaries, as well as inshore and open waters. The laboratories working on this are ASTI with NERA. They're looking at sea lamprey, European sturgeon and shad. And Phil Davison in CFAS is looking at smelt and the thin-lipped mullet. The second analysis I want to talk about is our Shad Microchemistry. The samples collected have been sent to Francois in the INRA lab, and we are looking at otoliths and scales, looking at the elemental signature through the life history of the fish. Very similar to the rings of a tree. The outer ring will represent water where the fish was caught, and the center will represent the natal region or the juvenile waters of the fish. Analysis along the length of the otolith could capture water signals from various different river water bodies showing movement of the shad along our coastline. We are also looking at the microchemistry of the thin-lipped mullet. This is a tool to examine the habitat use of fish, capturing the movement or multiple movements from marine waters into estuarine waters. The Untitled Microchemistry will complement the tracking data being collected in many of our case study locations. The final new analysis is a genetic study looking at the dynamics of shad hybridisation. We cannot identify shad hybrids visually after the first generation, but a study by Théobois et al. 2019 found a mix of shad species and shad hybrid generations along the French coastline. Our samples will be a mix of marine samples and case study locations. They will be adult and juvenile samples, and they will contain tissue and/or scales. The samples used for genetics will also be used in the microchemistry analysis that we discussed earlier, and this will help us fully understand the dynamics and the connectivity of shad populations around our case study locations. To finish up, I wanted to show you our timeframe for the data collection. The data collection is complete for eDNA ICP-MS and the shad hybridisation. The lab analysis is being undertaken as we speak and will continue until end of January 2021. And the data interpretation will take place in the first half of 2021. To give you an example of some of the other data we're collecting, as part of WP6, we're looking at gathering bycatch data, tracking data, this is telemetry studies, and distribution data, which is taking advantage of other programs and surveys such as the Water Framework Directive. We will transfer all relevant data, such as data to WP 5.2 around September 2021, and the economics data will go to WP 4 in the end of January 2021. Just to highlight the impact COVID-19 has had on our year, the lockdown and restrictions have impacted on field surveys and data collection. For example, the shad spawning would have taken place in May 2020, and this was missed by a number of our teams. However, our partners have managed to maintain sample size by using existing samples from their institutes. This is to ensure that we keep to the deadlines and reach our deliverables. Restrictions have also affected access to laboratories, and this has delayed analysis and access to scale samples. On behalf of Willie Roach and myself, and all the people involved in WP6, which as you can see here, there are many, I'd like to thank you for listening.